Hello everyone and welcome to the nanomedicine discussion on clinical trials. So first let's talk about design differences between imaging agents and therapeutic agents. So far we've talked about imaging agents or therapeutic agents in isolation, but since this is an engineering class we should really talk about the design and the design differences. So on the dosing for an imaging agent, it really should be a single dose. You don't want to give multiple doses of an imaging agent because tumor to background ratios or tissue to background ratios or target to background ratios, however you want to say TBR, are very stringent. So you want a high amount in the target tissue and a very low amount in the background. You have to be able to see what it is you're looking for. Therapeutic agents, on the other hand, are designed to have multiple doses because that is a pharmacodynamic response. There's a therapeutic effect, and so most of the time a single dose is not good enough. The amount for an imaging agent should be tracer amounts, so very, very small amounts for imaging, and in fact, there should be no effect on the target. Where in therapeutic agents, you can give large quantities of them, and that's why the FDA, getting it through the FDA is more difficult in a therapeutic agent than typically is for an imaging agent. There is no tolerance for any kind of toxicity with an imaging agent because you're just trying to see something. You are not trying to cure something, and so no toxicity at all. Where therapeutic agents, cancer is one of the best examples. There are significant adverse side effects to most therapeutic agents for cancer, chemotherapeutics, but um, that's better than dying from the cancer. And so, again, if you, although have an antibiotic infection or antibiotic, if you have an infectious disease, for example, strep, well, there's a little bit less tolerance because the antibiotics do quite well. So is there an effect on target for an imaging agent? That is no. For the therapeutic agent, yes, absolutely. You have to be able to have some kind of response. The target to background requirement is stringent for an imaging agent. You have to be able to see it. It's irrelevant for a therapeutic agent, except for in that toxicity. So if there's a lot in the background or a tissue that has high toxicity with the drug, then that's going to be a problem. The effect or the timing, it should be minutes to hours for an imaging agent. Therapeutic agent, it can be minutes, hours, but days to months because you really want the therapy to be sustained, depending on what you're treating. Marker of success, so this is our gold standard, that's histology for imaging agents. You want to make sure your target is there. Therapeutic agents, it's survival, symptoms, other things. And then preferred targets for imaging agents are receptors, enzymes, extracellular matrix, endothelium, or host response. Therapeutic agents is anything you can build a drug against. So let's first talk about clinical trials and why we need to do them. So the first question is, who is this? So what happened was in Europe, thalidomide, this compound was approved for distribution in Europe. However, at the FDA here in the United States, this is one of their many success stories, where looking at the data, one of the people decided that this was not a very good agent and stopped it from being approved in the U.S. And so then what happened as moms who were taking it because of morning sickness, their children were born and they had what was called phocomelia, and so their arms were more like flippers. And that's because thalidomide exists as a racemic mixture. One of the racemers is very good and it does what it's supposed to. The other racemer is what caused the birth defects. And so the process of the FDA and FDA approval, while it takes a bad rap for delaying drugs being on the market, so on and so forth, it's actually there to provide, to make sure that patients are not being harmed. So who was that? It's Frances Oldham Kelsey, and she was one of the staunch opponents of thalidomide being FDA approved. What she said was the clinical trial reports were more on the nature of testimonials rather than the results of well-designed, well-executed studies. And that's exactly what we do now. These trials have to be well-designed, well-executed. So bench to bedside, this is a terminology that's thrown about a lot. And so what this means is that first we have extensive laboratory studies in human cells and then also in animals. You are required to do some animal testing. The data is sent to the FDA for approval, so you can continue to research, or you can actually research in humans. You have to get what is called an investigational new drug, or an IND. And then human testing can be conducted. It's conducted in 4 phases, each of which is its own clinical trial. Data is submitted to the FDA after each trial, and approval must be obtained to continue. And in part of that approval process is, for example, all the studies done in the animal work, the toxicology work, so showing that it's not toxic, even sometimes 100 times the dose that you would actually administer to a human. Manufacturing data, how is it manufactured? How sterile is it? What's its stability? What's its mechanism of action? All of these things have to be submitted and written to the FDA. Also, you have to have someone write a protocol. So what is that clinical trial going to look like? How many subjects? What's the dosing? All of those kind of things have to be looked at and approved by the FDA before you can start dosing patients. So what are clinical trials? Clinical trials are medical research study involving people. So this is exactly for cancer, but it's very common in other types of pathologies. But clinical trials will look at preventing cancer, so using drugs, vitamin, or diet to reduce risk, or not smoking. Screening would be a trial, so screening for people at higher than average risk or for the general population. Diagnosing cancer, treatment, so new drugs, combination of drugs, new ways of giving treatment, new types of treatment. And then controlling symptoms, so new drugs or complementary therapies. Why do we have to have clinical trials? It's to find out if a new treatment or procedure is safe. Really important that it is safe. What the side effects are. It has to work better than the currently used treatment, and it has to help you feel better. So new treatments have to be thoroughly tested. And there's different types of trials. So there are 4 phases of clinical trial. Each treatment being tested has to go through all these phases before it can be used. So phase 1 is looking at whether a trial treatment is safe or has any harmful side effects. The research team will also find out the best dose to use. This is the maximally tolerated dose, so the dose keeps escalating until the patients no longer can withstand the treatment. A phase 2 trial will look at how well a treatment works. Only a treatment that has got through these 2 phases goes into phase 3 testing. Phase 3 trials will test a new treatment against the existing standard treatment. If it gives better results, it may become the new standard of treatment. And then phase 4 trials are the only trials that are carried out after a drug has been approved. Here they collect information about side effects, safety, and the long-term risks and benefits of a drug. So phase 1 trials, these are the earliest trials in the life of a new drug or treatment. Phase 1 is written as phase 1, so whether it's capitalized or not. They are small trials recruiting anywhere up to about 30 patients, although often a lot less. So pay attention to the numbers of these patients, about 30 patients. The trial may be open to people with any type of cancer, or it can be very specific depending on what kind of drug you're testing. When laboratory testing shows that a new treatment might help cancer, phase 1 trials are done to find out the safe dose range. So you really need to know what your therapeutic window is, the side effects, how the body copes with the drug, so that's the pharmacokinetics, and if the treatment shrinks the cancer. Patients are recruited very slowly onto phase 1 trials. So although they don't recruit many patients, they can take a long time to complete. And that's because you want to wait in between treatment for 2 to 3 weeks, sometimes even longer, to see what kind of adverse events might happen. It might not happen at the very beginning. It might take a few weeks for some kind of adverse event to happen. And so you don't want to just dose all the patients at once. The first few patients to take part are called a cohort, and they're given a very small dose of the drug. If that goes well, the next group have a slightly higher dose. The dose is gradually increased with each group, and then the researchers monitor the effect of drug until they find the best dose to give, and this is a dose escalation study. In a phase 1 trial, there's lots of blood tests, so that way the researchers can look at the pharmacokinetics. They also look at the metabolism, so how the body gets rid of the drug, and they will record any side effects. People that usually take part in phase 1 trials often have advanced cancer, and this is usually they've had all the treatment available to them. They could benefit from the new trial, but they probably won't. It really is looking at and aim to look at doses and side effects, so this work really has to be done. Phase 1 trials are very important. The other part of clinical trials is informed consent, and so you cannot do any kind of clinical trial or experimentation on humans without informed consent. And this means telling them the benefits and the risks of potentially getting this drug. And in a phase 1 trial, it may be that they don't benefit from this at all. And the informed consent papers have to be written for someone with a 6th grade education. So they can't be in technical jargon. They really have to be able to understand it. A phase 2 trial. So not all treatments in a phase 1 trial will make it to phase 2. These trials may be done on people who all have the same type of cancer, several different types of cancer. It just depends on, again, what drug. Phase 2 trials try to find out if the new treatment works well enough to test in a larger phase 3, which types of cancer the treatment works for, more about the side effects and how that could be managed, and more about the best dose to use. Although they have been tested in phase 1 trials, there still may be side effects that the doctors don't know about. Drugs can affect people in different ways, and in a phase 2 trial, there are larger groups of patients than in a phase 1. There could be up to 100 people or so taking part. So if there's a side effect that's a low event, but it's a highly toxic side effect, then more and more patients will actually allow you to uncover that potential adverse event. So if the results of the phase 2 trial show that the treatment may be good, then it moves into phase 3. Most trials fail at the phase 3, and these phase 3 trials compare new treatments with the best currently available treatment, the standard of care. These trials compare a completely new treatment with the standard treatment, different doses or ways of giving a standard treatment, a new radiotherapy schedule with the standard one. So phase 3 trials involve more patients than phase 1 or 2. This is because differences in success rates may be small, so the trial needs many patients to reach statistical significance. So for example, if 6 out of 100 people get a remission with a new treatment compared to standard treatment, if there were 50 people in the new treatment group and 50 people in the standard treatment group, there may be 3 more people in remission in the new treatment group. The two groups would not look that different. But if the researchers gave each treatment to 5,000 people, there could be 300 more remissions in the new treatment group. And sometimes phase 3 trials, and often phase 3 trials, involve thousands of patients in many different hospitals in different countries. Most trials in phase 3 are randomized. That means the researchers put the people taking part into 2 groups at random. One group gets the new treatment and the other the standard treatment. And then finally, phase 4 trials. Phase 4 trials are done after a drug has been shown to work and has been granted a license or approval. These trials look at drugs that are already available for doctors to prescribe rather than new drugs that are still being developed. The main reasons to do a phase 4 trial is to understand more about the side effects and safety of the drug. This is being given to many, many more people. You can study these on long term. So what are the long-term risks and benefits, and how well the drug works when it's used more widely than in clinical trials. So that's the extent of the clinical trials that we have, or the clinical trial system that we have here in the U.S.